Nice group this morning. Can I start off by first saying thank you for coming here. Colorado obviously is a very different venue than Florida has been, but hasn't this been a remarkable week? The timing of me being able to address you today is absolutely deliberate by waiting until times like now, especially coming from Florida. So the future's bright, and we'll conclude when I'm speaking later in the talk about just how bright that future is, where the science is going, and it's not only about the plant. It's actually about the receptor system in each of you and myself that has defined itself through discoveries through the plant. So first of all, so I know who's out here, it's a great crowd. Who literally is in healthcare? Who's healthcare? Can I just see a show of hands? That helps me when I see who I'm speaking with. Okay, how about any homeopaths? Naturopaths? Osteopaths? Allopaths, MDs. No, people don't know that word. Okay, so we have a real good distribution between different healers, okay? Nurses? And finally, but to me, very importantly, plantsmen and women. Who are plant people? Okay? And finally, the last group, people in the processing of the plant's chemicals. We got some of everybody here. And that's the beauty of when you hold something in a city like Denver. What I'd like to do then is really just get through some of the initial parts, because I'm going to try to share with you over the next several minutes, not just where the future's going, but I teach medical students, and I emphasize that if you want to know where you're going, you've really got to know where you're at. And in a grand scheme of things, if you want to know where you're at, it really does help to look and see where you came from. So when people just see where we came from, and you see a political movement, that's very short-sighted. The fact is, we came from very, very different backgrounds that's not simply 10,000 years ago, but endocannabinoids evolved far before that. These are the absolutely essential disclaimers, obviously attestations, and by the way, anyone who speaks to these always knows that there's always a statement there that says, anytime you speak about something that is off schedule, you need to let people know. Everything I talk about today is off schedule, because it's Schedule 1. So moving along then, I'd like to be able to leave you by the end of today's talk with a feeling that you have a different understanding, because I think that as research gets out there, there's a big disconnect between clinicians and the researchers. The only reason I partly understand what the researchers are doing is that's my background from before I went into clinical medicine. This is a huge field. It's incredibly complex. It's being revealed that way in its complexity every day. And that's not a confounder. That's wonderful. That shows why we're all finding slightly different slants on it. So I want to try to keep this in perspective by first showing you something that may look like a little nerdy slide there. This shows evolution, and not just evolution of 10,000 years, certainly not just the last 35, and not from the point of the Marijuana Tax Act in 1937, but going back and way, way back, because the endocannabinoid receptor system dates back not even to vertebrates, to invertebrates. And so we're talking about the difference between 500 million years, the B, between when the first endocannabinoids were used in animals, early animals, actually sea squirts, they came out of Tampa Bay, that's where the research came out, to about 60 million years ago when the plant first appeared. Humans' use of the plants, though, goes back about 10,000 years, and this is part of the perspectives I want to bring about really where it started, so you understand more than anything else that there were a few places in the world. You had areas which are listed in red here and up towards Europe here, which were called refugia. These were the places that plants and animals found it warm enough in the last ice age, Pleistocene, to survive. And that's where human beings likely first encountered cannabis. And from that time, we dragged it all the way around the world as a medicine. This, in the interim, about 3,000 plus years ago, was showing burial tombs. And in those burial tombs were actually not just some cannabis, but female cannabis flowers. So early humans may have known something about it, and certainly by the time of having recorded history, medicines that go back about 5,000 years, human beings knew what secrets may have lied in those female cannabis plants, because from that point, we drag the plant around with us everywhere in the world. And I would rather give credibility to 10,000 years of human use to figure that at least teleologically, it likely works favorably with our genes. The last 30 years or so, we have changed the plant markedly to no longer have the normal balance between different cannabinoids, much less the terpenes, because the parent plants were different than this. This is what you see today. And those capitate glandular trichomes, all the things that leave that little white fuzzy stuff to the rich flowers you see on the tips, that's where the chemicals are made. And it's not just the cannabinoids or plant-derived cannabinoids, they're the terpenes also. And today's plant is noticeably different at 20% or so THC content versus 1970s of 5%. That's big and that's important because dominant genetics have pushed the plant that way, and at the same time have left CBD, of course, that's the most familiar other principal phytocannabinoid, in considerably lower amounts. So what does that mean? We have a more potent plant, and if the one thing out there that's pretty obvious about the plant potency derives that, hey, if you're going to tell one thing to somebody more than anything else, be careful. Because no matter what your effects and tolerance may be from it, if you can't control your dose is going to control you. There's case in point, isn't it? Okay? And that's 1978. The plant was very different then. But the enthusiasm around it, and frankly, any of you that may have seen this movie who know this scene, what Cheech does with the paranoia, the consequences that happens, hysterical. But this is one of the problems that's been exemplified and absolutely expanded with today's plant. So what do you have to do to try to find out in our past what the plant probably had? First of all, you need to find very few places that still exist in the world where the plant has been grown for thousands of years. The Bekaa Valley in Lebanon happens to be one of them. And when you look at the plants that are coming from there, their balance of chemicals is very different. The plants that would be typical of the plants all the way on the right side of this slide, mostly if not exclusively THC, yeah, there's side effects in that, as compared to all the way to the left part of the slide here, which shows CBD. That's otherwise basically hemp. But cannabidiol did exist in older cultivars, landrace plants, more towards what's represented in the middle of this picture, in approximately a 1-to-1 ratio in certain places when you go back and try to find them. And it's not an accident that some major pharmaceutical companies dealing with the plant's formulary for multiple sclerosis deal with plant constituents from the cannabinoids that are approximately in that ratio. So this is what humans lived with for 10,000 years, not necessarily what's out there today. What I want to do now is jump right to 2016. Here we are. And those of you that are clinicians or see patients, I want to share with you something that was published 3 years ago in the New England Journal of Medicine. Now, this is a case study, and I'd like you to read it. You can read it with me, you can read it yourself, but suffice it to say, it's a lady who's 68 years old, has metastatic breast disease, and she's complaining about different things associated with what she's got. So go ahead and read it. She's getting chemotherapy, she has low energy, minimal appetite, a lot of pain in her lumbar and thoracic spine. And so she goes on to saying what she uses, and she says, well, you know what, she takes onestron, to basically Zofran, she uses some compazine, things like that, with minimal success. And at the same time, she takes a lot of acetaminophen, Tylenol, and sometimes oxycodone at night, opiates. And so she comes with a very typical question that your patients bring when they're getting older and sick like this and not doing well. She wants to know about what can she get to help her with her nausea, her pain, and her fatigue. Remember, she's asking that. And she lives in a state where you can get this. So from seeing that there, can I see a show of hands? Who thinks that she should get cannabis immediately, and that's what you should do to help her? Anybody raise your hand if you think that she should get something else, or something else about her case that you'd want to know about at this point to help her through the problem? All right, many people put your hands up, and I want to be constructively critical, because obviously she's going there and she knows something. Yeah, some of the things that she's complaining about likely will be helped from cannabis, in particular the pain. But remember, she's a lady who has metastatic cancer and she's coming to see you as her primary care doc. She's got mets to her spine. It's not even where her primary is. She needs radiation to that spine to minimize the process, or it's just going to keep happening. So she needs the radiation, maybe even first, Some of her symptoms are probably coming from the narcotics she's taking, probably could do better with some cannabis there, and maybe the nausea is coming from that. The fatigue is her cancer. Let's see what you can do to arrest her cancer from continuously to invade her nerves, and that's a help too. So the message here is at the end of 2016 for evidence-based medicine, which is what the speaker before me was emphasizing, this pretty much across the country right now are regarded as the best evidence-based diagnoses for which we have for cannabis. Understanding that it's still not a first-line medication for anything. It's in addition to other things. So in addition for the lady Marilyn in the last case who needs some radiation to her spine and to treat her as a whole human being, cannabis may be helpful because, look, she's got that neuropathic pain. She's got cancer that's eating away at the nerves. It could likely help her. The chemotherapy, nausea and vomiting wasn't from cisplatin. It was from one that doesn't tend to cause it as much, so that probably was a little red herring in there that was meant perhaps to throw you as clinicians. But certainly muscle spasms from MS would qualify, lower urinary tract symptoms, and obviously the nausea and vomiting of anorexia, HIV, neuropathies. They are certainly having a benefit of having evidence-based data to support it, while there are other things that are pending. And even some that may not be in that big list that your previous speaker showed you of how many diseases sort of get piled on. I'll share with you, I have glaucoma. And glaucoma sort of has been grandfathered in because you know what? It does reduce intraocular pressure. But not so well. It doesn't have yet the delivery vehicle to bring it, because it's lipophilic, into the eye to exert the effect. I come from Miami, where we have today the only still surviving IND patient who has glaucoma who received it for the government, and she's a 1 in 10,000 case for glaucoma. So eye drops are still the mainstay in treatment because it doesn't carry not only the side effects, but it's more efficient, covers more types of glaucoma, and it absolutely is something of greater duration than cannabis if it were to be used for glaucoma. So at this point, and it's not meant to be a culture shock, it's the reality of everything else is anecdotal, but at this time, there's a tremendous amount of research going on right now around the country and around the world, and new things are being revealed all the time. So this is where we stand in 2016. This is what you have available. You right now, of course, have cannabis in different states. The middle two chemicals there that are Schedules 2 and 3 is not cannabis. It's not even a balance of medicines. And in fact, what happens when you extract single chemicals are known to be not particularly as well effective as the balance of cannabinoids, especially things like Marinol. The bottom two are things right now that are in their phase 3 or currently under the FDA's look, and those may be around next year. But in the meantime, that's what we're stuck with today in total formulary availability. So what does that mean? I want to approach this now going back to understand how we're wired to work with plant chemicals. The endocannabinoid system is there through evolution of millions, if not 500 million years, such that the chemicals that we find in plants are actually defenses for the plants themselves. And this isn't even talking about the plant, which usually you're talking about plant alkaloids, I'll mention in a moment. This is how similar we are to a plant, to the very lifeblood that we have, hemoglobin, compared to chlorophyll in a plant. The molecules are identical except for a 2+ cation in the middle, and physicians use iron the same way a farmer would use magnesium to help a plant that's a little sick. Optimize hemoglobin, the plant does better. Optimize hemoglobin, the patient does better. So what does that mean as far as how people have understood plants for thousands, if not tens of thousands of years? Civilization's earliest healers had to understand nature's resources. Even Hippocrates said nature itself is the best physician. And if you think about it, even before people like this, primitive tribes, if you had a healer in that tribe, that healer would have to be really good with knowing how to sustain a plant. If they couldn't, they would have lost their formulary, and then they're not regarded as a good plant person or a healer. So plants and people have evolved for thousands and thousands of years. When you look at the traditional healer's way of looking at combinations of plant chemicals, which is what was missing in the Marinol and the Sesamet on the previous slide, people who practice Eastern medicine, who would never dream of giving a patient one herb, know that combinations of things are in fact what helps people survive. And here Deepak Chopra says it better than I ever could. So what happened then in modern science? How did we evolve, and where did we come from more recently? Well, in 1947, the Nobel Prize in Chemistry went to Sir Robert Robertson. And he discovered alkaloid chemistry. Alkaloids are those chemicals generally in plants that the plant uses as defenses against herbivores. So animals don't go and eat them. So most of them taste bitter. That's why most medicines traditionally taste bitter. And what he did his early work in the 19-teens was finding the precursors to cocaine. But by the time he's a ripe old man in his 80s, which most octogenarians get their Nobel Prizes then he was known for cocaine, and in particular because his acceptance speech for his award came with a quote, and I could paraphrase it, but he basically says that those chemicals that are in plants that are known in prehistoric cultures often carry with them the use that prehistoric people bring with them. And in cultures that have those medicines to date, knowledge of their use often goes back to those prehistoric times. So other types of work outside of botanical medicines, many of which are pictured here, I think Martha mentioned coffee before, chocolate, certainly tobacco. These are all plant-based alkaloids, and cocaine and opium. These are all flower-derived alkaloids. Cannabis is different. It's not an alkaloid, and the medicines that come from it are not. But botanical medicines, nevertheless, are the mainstay of how we help and cure people. Cannabis is different, and we owe a lot of thanks to Raphael Mechoulam, because the history of this is barely 50 or so years old. And in that brief period of time, this is what we've learned that makes cannabis so different. Remember that this is a science barely in its infancy. So it would be really nice to move forward fast because you sort of have this intuitive feel from anecdotal cultural use. I don't put down anecdotal or cultural use. It's a great system for seeing what secrets may lie there. But as Jack brought up in the speaker before me, that's not evidence-based data. The plural of anecdotes is not data. So where do we go from the fancy science? There's not going to be a test on this after, don't worry. But the same parent compounds that derive the cannabinoids also derive the terpenes. This is very important from a biochemistry perspective. And I find that this is one of the major contributions, of course, that Professor Mechoulam leaves. But then, more important than that to me as a clinician was the discovery a good 20 or so more years later about the receptors themselves. It's a beautiful slide from Phytex. It actually shows a flayed open synapse, the presynaptic side and the postsynaptic side. Suffice it to say that if you think about what the endocannabinoid system is doing, it's functioning as a feedback loop such that the release of chemicals, and we're not only talking, cannabinoids now, we're talking what the synapse on the presynaptic side releases. Everything. Dopamine, serotonin, any of the things that you can think of that are released have to have a way of self-regulating. Now, I'm going to try to present this not from complicated slides that these last two, but in a simple way. And the simple way would be to avoid words like homeostatic modulation. That to me is, what are they talking about? The only reason I know it is from my specialty background is I say that in front of somebody, they go, what? So here's what's happening. Down here at the bottom of the slide is where you see on the postsynaptic side, the body's own ligands, the body's own cannabinoids. Anandamide and 2-AG, again, Professor Mechoulam's contributions. They're different than most other neurotransmitters. They go retrograde back to the presynaptic side and interact with the CB1 receptor that you see over here. The CB1 receptor then is what's responsible for changing the outflow of what things are released from that neurotransmitter released from the presynaptic side. So this becomes a self-regulating loop that's within all of our synapses and basically all parts of our body. And I listed the CB1 receptor here, but there's CB2 receptors also, we'll explain in just a moment. The reason why you see the arrow all the way on the right here of THC is because THC is mimicking what the body's own cannabinoids of anandamide and 2-AG are doing. That's how it works. That's the magic involved. So to take it one step further, this is not something that I really want you to appreciate, or you don't have to go to town with this, but this is shown, and this slide happens to come from GW Pharmaceuticals, of the varied effects that changing how that receptor works changes the release of chemicals. So you have things that are called positive or negative regulation, partial, total, agonists, antagonists. This is all the subtleties of receptor-specific pharmacology. And it works in infinitely detailed ways, constantly in every one of us. That's it for the nerdy slides. So let's get into something now, though, because the two little green dots off to the left, all the way on top, those are the CB1 and CB2 receptors. Well, as the end of 2013, those are all other receptor systems, and there's more now, and those cannabinoids affect potentially every single other receptor in the body and can affect potentially all other forms. So those receptors that we think of for cannabinoids sort of were identified in a similar way by the opium poppy. That flower, but many years before, in the 1970s, were found to also work through an endogenous or receptor system we have in all of us for the endorphin system. In a sense, they both help pain, but they do so in different ways. The problem with the opiates, as you know, is this. And this was published 2.5 years ago, before Florida had its first law. I will tell you I'm wearing a little badge here from the Florida Medical Association, but that doc to me looks uneducated and frankly bored, and the lady there could be my mom. And it's a very legitimate question she's asking. But when you prescribe narcotics, it absolutely has problems. The CDC recognized this. For all of you that may be primary practitioners, care practitioners, you might have received these guidelines because once there was a push a decade or more ago about controlling pain, doctors did things fairly irresponsibly. Big Pharma probably jumped into it with the opportunity, and now look at the mess we've created. So taking it from there then, what's the right thing to do? It becomes really an ethical discussion, and I'll have it briefly with you because those of you that are practitioners, you want to do the right thing by your patients. And ultimately it comes down to, should a physician offer compassionate access to cannabis while it's still under prohibition? Now, I'm not saying that we don't have people here from states that are or are not legal. That's not the question. The question is, it's still federally prohibited, so what should you do to do the right thing? Well, the American Medical Association absolutely has a stance on issues like this, and what it says in Article 3 is they say a physician shall respect the law and also recognize the responsibility to seek changes in those requirements which are contrary to the best interests of the patient. Sounds nice, right? Totally ineffectual, and here's why. Here's why. Mainstream docs have two licenses. It's not only the license in the state you may be practicing, whether it's permissive or not, but you have another license, your DEA license, the license from the federal government that allows you to prescribe medications. Mainstream docs are not about to see a patient and authorize them to get cannabis, even in states where it's legal, out of fear of losing their tool bag. That's what that DEA license is. So that's a huge confounder, and it's a great impediment until the scheduling of cannabis changes. But if this isn't effective, then there's really another reason why I want you to understand that mainstream docs are not part of this. And that's because most states that so far have passed legislation, including where many of you come from, often are doing it by saying which plant chemicals can be used for which diseases. You saw big, huge lists of which diseases, and we're critical of states that have CBD only, and now they add THC, and very often in different percentages. Well, the last time I looked, saying what medicines could be used for what diseases, it was called the practice of medicine. And physicians hate to be legislated the practice of their craft. Let the people who do that go ahead and get licensed and go to school and learn this. In the meantime, you get a serious pushback from mainstream medicine about this, and it's not hard to understand why. So there's a different approach. And I'll tell you that one of the things I've done that actually has kept me from sort of coming out in this arena is because I'm the immediate past president of our medical school in Miami's Alumni Association, where I'm there giving the oath of medicine to 200 new docs every year and telling them what the right thing to do is. And I look, absolutely, there's Hippocrates in the bottom again. Nature itself is the best physician. I get it. So there's a different way of thinking about what to do in this arena, and it's not medical legal ethics. It's the same basic question, but it reduces to a different approach, and that approach is called bioethics. And I don't know who here is familiar with what bioethical processes are, but let's just say it's a science in ethics that has evolved since the end of World War II, when modern science presents us with things we never had to really deal with before, controversies. This is the same process a decade ago that stem cell research had to derive from, when originally having a very limited stem cell line that researchers knew hey, we're not going to go ahead and genetically modify and make some kind of weird human being. This is about using what we need to do, the tools to help people. And today, stem cells save lives every day. That process of bringing about the new science is lacking so far in endocannabinoid science to make it acceptable. So this overall could be a statement that would be not necessarily a compromise. This is not a Jeffersonian effort, so you bring two controversial sides together, but basically irrefutable statements about the way things are. Human beings are instinctual creatures, and they will use medicines in whatever way they have to in an effort to alleviate stress and pain. And it's not always physical. It could be psychological and even spiritual. So that's from the patient's perspective. From the healer's perspective, get out of the way and let us take care of the patients. That's why we're here, and that's what we want to do, provide humane relief. That's the way you answer the question of providing compassionate care while still under federal prohibition. So, in a sense then, what's happened is this, and there is a sense that you get things backwards, and maybe there are stakeholders in this who are forcing us as healers to think of things through a skewed lens. You know, the words perspective and bias, they really are almost synonymous, aren't they? But just with different levels of cynicism. So I don't care if you want to call yourself biased or having unique perspectives, but mine is that the science and the ethics need to be leading, not the other way around. So what happens then when the government comes out and says, as they did in August, again, we're going to keep it Schedule 1 for now. And the DEA, upon making that announcement, those of you that may follow JAMA, the Journal of the American Medical Association, may have seen this piece. It was an editorial piece written by this Federation of State Medical Boards. All physicians strive to be board certified if they can in whatever they choose to practice, the highest standard of their formal education. And the Federation of State Medical Boards, covering all 50 states and commonwealths, came up with 10 practice guidelines that should be expected of physicians or people who are authorizing patients to get cannabis, even in states where it's legal, while it's still federally prohibited. And the very first statement was, right here, they say a cautionary note saying advising physicians, actively licensed physicians, to abstain from using marijuana while practicing medicine. It also goes on to say abstain from authorizing your family members to get it, and then it gives 9 other guidelines to show a bona fide doctor-patient relationship. These kinds of guidelines have already been introduced to certain states, including California, and I anticipate that ultimately, upon the rescheduling of cannabis federally, that this type of model will be shared, because we are expected to behave at a higher standard. The same way you wouldn't find great tolerance for a law enforcement officer breaking the law. It's where things are today politically, legislatively, to say, well, if you're going to go there, watch out and make sure you're clean. So what I want to do now is I want to try to tell you how I approach seeing patients, or especially teaching students to deal with all of these subtleties of what the plant can bring, because through early research, in this case, Vincenzo Di Marzo, in 1998, what he said was that relaxing, eating, sleeping, forgetting, and protecting were some of the messages the endocannabinoid system is relaying. And so I want to teach you briefly this morning now, from birth right through death, what this means. By the way, Di Marzo is still the most frequently cited researcher of all time. So even though this statement comes from 1998, that's been the way I've thought of things since then, and I'd like to share with you a model for how those 5 elements, relaxing, eating, sleeping, forgetting, and protecting, can be applied to your patient care through 2 receptor systems, and they're very different. We know a lot more about CB1 receptors than CB2. CB2, of course, immune system. The brain with CB1 is obvious, and that's where most people regard it currently today. But when you look at endocannabinoid science, especially in context with what the National Federation of State Medical Boards said, boy, that plant is distracting, isn't it? And if you're a pure researcher, even a pure clinician, and you're trying to shoot bullseyes, the moment as an archer you know that your gaze, even your thought, shifts to the arrow in this analogy, which could be the plant, you're not really hitting the bullseye as much, are you? So this is really of the statement of how to target this big receptor system and do it safely and efficiently. So targeting the endocannabinoid system is the subtitle of this Sentinel work paper that came out, again, almost 3 years ago by the NIH. Two NIH very highly respected researchers, okay, Cal Packer and George Kunos, they're both MD PhDs. Okay, they're not only your PhD researchers who know a lot about what cannabis does in a petri dish or to a lab rat. I'm a clinician. As soon as I really care what it does to a lab rat, I'll let you know. In the meantime, people are what this is about. And when they can come up with a statement that says modulating the endocannabinoid system may have therapeutic potential in almost all diseases affecting humans, wow. That's a huge statement. That tells you how far-reaching this can be. We're not there yet. At 25 years science, we don't really know it yet. So what I'd like to do is share with you this basic model. And you see around the outside the relax, eat, sleep, forget, and protect. But what I've done is I've worked the CB1s on the top and the CB2 on the bottom, because I think when you see it the way I'm going to present it here, it may make more sense. And this is the way I like to present when I teach students about looking at the endocannabinoid system, and most importantly, still as a target. A very well-focused target that has nothing really to do about the plant, but when you see the way that it's presented, I think you'll see how people use the plant the way they do, and not babies through birth through death. It's a model to show you that whether your patients coming in are young or old, at the very beginning or the very end of their life, their endocannabinoid systems are working to help sustain them. So here's where it comes from. If you look at what the endocannabinoid system is, forget the words modulating and homeostatic regulation. It's simple. In terms of how it really works to keep you healthy, it sustains an organism by adjusting its physiology. Whether it's a sea squirt from 500 million years ago, or the highest evolved, if you think we're that, human beings today. And here's how the individual components of those things work through CB1. CB1, we said, involves relaxing, eating, sleeping, and forgetting, while CB2 is really the part to me that's going to be even more fascinating for protecting. Now let's take it like from that birth through death model that I was saying a moment. CB1, in terms of how it works, really what do babies do a lot when they're born? Yeah, they eat and sleep. And what about mommy? So in fact, CB1 receptors are essential early in life, reproduction, health. When we're growing, they're absolutely essential. Growth is from eating and sleeping. You know that, we know it, it's intuitive. Think of the endocannabinoid system as intimately involved in that way, and you'll have the first part of your patient's life here, so you'll get it. But what about when we get a little older? Stop growing, we really actually stop growing in your late teens and early 20s. And as sad as it may sound, from the point when you stop growing, you slowly start to die. It's slow and gradual, but the definition of growth is decay. So in that sense, as we continue to age, and through most of our adult life, we do things to help us relax and forget as ways of dealing with stress. Doesn't only have to be physical, and you know this. But when you're thinking about relaxing and how people, through their CB1 receptors, what they do, most people who found that they use pot to chill out are doing just this. Doesn't have to be from pot, but it's an essential part of how the endocannabinoid system works within us. And maybe the most fascinating part of all is the forgetting part, because that's really what adults do as they experience those things in life that represent stresses. And a stress doesn't only have to be pain. A stress really can be not just pain, but even pleasure. So when you're looking at what parts of the brain are involved in this, it's largely hippocampus, okay, and the amygdala. And those are the parts in the bottom left there. So this part of the brain where CB1 receptors are working are essential for you to understand why we forget. But most people don't understand why we forget. They think of patients or vets with PTSD, well, they need it to help them forget. But what about the way that human beings forget that's actually therapeutic? Why are we wired to forget that may have nothing to do with the plant? I'm going to propose to you Two little vignette stories here about pain and pleasure. Because in a construct, they're really both the ways that we're supposed to think sadomasochism, pain, pleasure. They present different things. And so let's first talk about pain. All right? Pain, obviously, if human beings could remember pain, there'd be no need in our language for the word brother and sister, because our mothers would have had one kid. All right, so there's some pretty obvious things about that, but what about this instead? Let's pretend it's 10,000 years ago, and a few of us are in some little tribe walking around trying to eke out an existence, to find a squirrel to eat or a fish, and out of nowhere comes some prehistoric wild hog, a beast. And I think prehistoric, like the size of a Volkswagen. So we're terrified, and we run, and we haven't eaten that day. We find a tree, we scamper up it. Maybe we're bleeding, our muscles are in spasm, and the sheer terror of that moment has our sympathetic nervous system going, our hearts pounding, eyes bulging. If we couldn't forget the sheer terror of that moment, to when nightfall comes, fall asleep and recover, when that pig's going home or something's going to eat it, we're going to be pig food in the morning. So the idea of being able, even acutely, to forget the sheer stress of that moment is essential. It's wired in us through this receptor system. And so human beings need to be able to forget for survival. Remember that analogy a minute, because what I want to do now is, let's switch to the pleasure side a minute and say the same primitive group, all right? And let's say it's Ice Age time. All right? So you have a group, and they're meandering like human beings must have done early in their days. And is it really healthy to remember the extremes of pleasure? Well, you didn't have pot then. You didn't have people shooting up or otherwise doing things, but we did have one thing that human beings have done for all time for pleasure: sex. And so what about if in that group of human beings walking around, you had two people in that group which were more wrapped up in it than anybody else in their whole tribe, and we'll call them Caveman Og and Lady Pleistocene. And they're always lagging behind and going at it, and everybody else in the tribe saying, come on, or find a cave somewhere. If you were a saber-toothed tiger and you wanted to pick the two to pick off first, who would you go for? In other words, they're not going to be around in the genetic pool with that kind of behavior by constantly remembering something and seeking out pleasure. So being able to forget the extremes of pain and even the extremes of pleasure is actually how we're hardwired through the receptor system of the endocannabinoids. That's really an important concept, because we think, oh, well, you're students, you need to remember everything, or remember every single thing I'm saying here. No, filter out, because every one of you has had different life experiences. The thing that makes this rich is that you may have had something that affects you that you find terrifying, that you will never do again, and so you're deathly afraid of it, you'll never do that. While somebody over here may have a tolerance for it, and somebody else may have a tolerance for something that is pleasurable that someone else doesn't fit within their gestalt. So what happens then? Those things that help us live in some healthy, non-extreme side becomes really the major determinants of your personality, isn't it? It's what makes each of us different from each other. And I'll tell you what, there's some really fascinating chemicals related to those chemicals in the body right now, in your brain, hundreds of them, and we don't even know why they're there. We know very little about the biochemistry of personality. This is a whole different field, but a lot of it is around the filter that we have for selective memory. So anyway, this is it then in a nutshell for CB1. Relax, eat, sleep, and forget. Protecting is really even more fascinating, because those are not primarily up in the brain. CB2 receptors are in your abdomen and generally immune system, and that's a part of your body which even the ancients understood was part of health and flow. So, in the same analogy, what happens with CB2 as you get older? You need it. Because the CB2 receptors, when they show up in response to diseases, and they show up exactly where you think they would, and they're there controlling things like inflammation, oxidative processes, things associated with immune function, but not the regular immune system which responds to protein invaders like bacteria and viruses. This is responding to different diseases that actually changes the physiology naturally to let you live with that disease and make it chronic. That's the way we generally live when we get older. CB2 receptors are very important that way. This shows a little bit of what effects it might do in the bowel, and all of those other diseases that get lumped in for hopefully diagnostics. This is where the secrets of CB2 research will hopefully come about. But in addition to that, look at this picture on the right. That's a PET scan. And that PET scan done in 2013, if you look very carefully, right behind the word life, you'll see a little red by an arm. That's an intravenous site where radioisotope labeled cannabinoids are injected and find their way to CB2 receptors. And it's not just where it's red, that's more blood flow. But if you look at the places, the yellow, the light green, and the pinks and the reds, that's where the CB2 receptors are, in the same area where the immune system is. And if you look at that distribution side by side with an ancient description of acupuncture points, and you see the density of acupuncture points, and those of you that know something about it, with chi flow, so much stuff going through the abdomen. Isn't it interesting how the ancients knew that that flow was essential to life, longevity, and health? So maybe, in fact, the endocannabinoid system is the missing link from gut-brain connections right down to seeing how they might have understood things in different cultures today. So where are we today? Well, the last few years, obviously, right here in Colorado, you had Sanjay Gupta doing his mea culpa saying, hey, you know what, the little kids with epilepsy, it seems that it really might help them. Jury's still out, but it's kind of exciting with the phase 3 studies that we're just about ready to hear. That's evidence-based medicine. So then in 2015, you had major journals, and I'm not talking about, Rags, Leafly, and other types of high times. We're talking about National Geographic and Time. 2016's even been better. And why? Because now open access data sharing, available to all of you the same way it is to me, has started not just with NIDA, but with other institutes within the National Institutes of Health, and this is free. It's available. You want to know what's out there? Go ahead and listen to it. A lot of them are pretty nerdy speakers, but they're smart. They may not have connected all the dots, but they're sharing what they have, because we're living in a day and age of global communications, and it's fast and accessible. So where does this put us coming up to 2017? Here's a study that came out less than a month ago, and this is really cool. This is the future. This shows, in the middle, if you see it, a receptor, and it's the receptor for CB1. And if you look very closely in the middle, there's a little green molecule that's sandwiched in there. That's meant to be the ligand, or what's bonding with the receptor. And on the outside, you can see different chemicals. To the top right, THC, and then you have anandamide and 2-AG on the right. Other experimental compounds, even rimonabant, which is not something successful, and some of the other JWH ligands that didn't do well. And even on top, you see one that's listed as AM6538. That's an experimental, lab-made ligand that was used to map out this particular diagram that really shows what the CB1 receptor looks like. So if you look very close in the middle, I'll enlarge it just a little. Can everybody see in there? There's a green molecule. The green actually are carbons. The blue is nitrogen. The little red dots are oxygen. Well, what that is, it's showing absolutely exactly where that ligand fits in a model of the receptor. This is done when you think about it, to identify a space where future molecules will be created that can fit in and not give you the total effects maybe of one response of CB1, but some partial ones. And if you look at this right here, that's the same molecule, and this is an actual picture. Okay, it looks like a caricature, but this is what it derives from. That purple net field representing the electron fields that are around where this receptor fits the key. It's a lock key thing. Very specific stereochemistry that goes into this. This is future, and this is cool, because that's not an artist's conception. This is actually what it looks like, scale and composition. So when you add a molecule here or take one away to make new compounds, this gives you at least a blueprint for what it might fit. But everything in science today is about something else: genetics. Genetics ushered in the 21st century, and I'll conclude with this slide because this is where things are going. I wasn't trained in genetics. I predate that. I was trained with receptor-specific pharmacology in the 70s and 80s, practiced that way. This is what the students now are getting. And it's not only human genetics, it's plant genetics. I don't know how many of you are aware, but the very first cannabis genome was done in 2011. Amandia Labs in Calgary. And we're talking still millions of dollars, several months to come up with the first genome that way. But that's a far cry from Arabidopsis in 2003, the first plant genome. Plants have been used genetically now with engineering to let corn give you greater yield by taking some sequencing out and putting a resistant feature in another crop or rice. But that's for plant crops. How about in medicines, though? The picture on the right shows you, as of the end of 2015, each of those lines is a different plant strain of cannabis. Over 150, I believe, have now had their genomes deciphered. And the common links where you see clustering there is absolutely what's shown, and that's the cool part, because there's a technology called CRISPR technology where you can extract certain sequencing of genes and put it in another plant and make that other plant express those genetics. So imagine this for a moment, when you can actually do that. We told you before that in ancient times you had these plants out there, and I can tell you that years ago there were people who were on what was called the hippie backpack trail looking for that heritage heirloom plant that they might have remembered from Vietnam, and lo and behold, they're really not there. They're kind of extinct like dodo birds. But they remember it, and different cultures remember it, not just where the landrace fields are or used to be, but it makes sense that if you could find certain cultivars that were ancient, now we're such a big melting pot that maybe there is one type that would work well for you because your ancestry derived from this part of the world and somebody's from another place. That's what they can tell you when they take a cheek swab now and give you a report back in a week to tell you your ancestors. It's pretty cool stuff. Apply that to plants now, and your search for the right cannabinoids and pulling them together with the global communication efficiency that's making 21st century science so exciting can really match the plant with human genomics. And then in addition to that, you now have the synergy of what I was saying before, open access data sharing. When you do that, you can maybe even realize global personalized medicine. And this might be what the plant identifies, but the process is timely. And here's why. Even though you can't take as much of a seed as bring it across a state line of cannabis, much less around the world with the plant, you know what you can actually share? Everybody's already got the plant around the world. So if everybody has the plant, and industrialized countries have new sequencing genetics equipment that can actually decipher this as efficiently as it's been done here in the United States this past year, what about when you can actually send the information across the world for the people to extract code? Because even though you can't transport that seed anywhere, you know what you can transport? The code, with the click of a mouse. So that, I think, is where the future of not just cannabis, but medicine goes with all of these accelerators. So I'd like to conclude with that because it's got great potential for a bright future, and I'll look forward to speaking with you at some time during the session.